Let us just wait for 1 more minute before we start. Nidish, you are there in the wrong class, I think. Nidish? Okay, still the crowd has not increased, but still we'll start the class. We were seeing about cell division. So yesterday we covered about prophase, metaphase, and anaphase. So today we'll try to go ahead with the telophase. So all these 4 stages are called as karyokinesis. What is karyokinesis? Karyokinesis is division of nucleus. Okay, so karyokinesis is what we call it as division of nucleus. So that is what we have been studying. So now we'll go for telophase. So yesterday we studied till anaphase, the cell was like this. So what was the happening? What was that happening? Basically, you have the chromosome is like kind of moving towards the pole. It has separated. Okay, so this is what would have happened till anaphase. So now when you're looking at telophase, what you will be able to see, you will be able to see 2 things. I mean, there are 2 different things that might happen when it is there in the early telophase. Okay, what you might be able to see is you might be able to see So what are you seeing here? You are able to see that this is early telophase. By the end of telophase, what would have happened? So this kind of situation would be the case. Okay. So now tell me what are the changes that you are observing? You are observing some changes. What are those changes that you are able to see here? Yeah, first thing, you are able to see that the nuclear membrane starts to appear. Okay. So the nuclear membrane is starting to appear. So that is something which you have seen. And then what are you seeing here? The chromosome loosens up. Yeah, this is all telophase. Okay. The chromosome decondenses to chromatin reticulum. Okay. So please copy this thing down. So this one was anaphase. This is information for thing. So this was anaphase. The diagram, this diagram is pertaining to anaphase, and these two diagrams are pertaining to telophase. Yes, mitochondria will be there, obviously. Please understand that the mitochondria will divide between the cells. As in like how the nucleus is going, in the same fashion mitochondria will also get divided. Okay, so you can write, continue writing. The chromosome is no longer visible. Okay, the nucleolus, Golgi body, endoplasmic reticulum, forms again, which means the organelles are starting to appear. So maybe like you can see by the end of it, you can see this kind of structure. Okay, so you should be able to see that the nucleolus is starting to appear. Earlier, those things and all got disappeared, no? So now they are starting to reappear. So that is also happening. This is like in the beginning of it, early telophase, but as it is progressing towards the end of the telophase, you might be able to see it. Will there be mitochondria? Yes, mitochondria might also be there. Okay, and then finally, a single cell now has two nucleus. Okay, so something that you need to understand here, as far as mitochondria and chloroplast is concerned, chloroplast or plastids is concerned, okay, they will undergo binary fission. What is that thing you should know? No, like they will not be like dissolved at all. So these two things are semi-autonomous structures, cell structures. What is the meaning of semi-autonomous cell structures? They will be capable of multiplying on their own. So which means when the cell is like when the nucleus is dividing like into two, what would have happened is like equal amounts of mitochondria would have gone here also and here also. And what would have happened from after the cell divides, this from here new mitochondria will start to form. Okay, so it is not like endoplasmic reticulum or it is not like Golgi body, which is like dependent on the DNA information. Okay, the information for endoplasmic reticulum development, nucleolus development, Golgi body development, all these things come from the DNA. Whereas in case of mitochondria and chloroplast, they can be forming right from whatever the information that they have. So it is like almost like how the cell is going to divide into two in the same way the mitochondria and chloroplast can get divided. Are we able to understand? So this is just a information that you should know. Mitochondria and chloroplast will be able to multiply like a normal cell. Okay, are you guys done? The parent mitochondria will not die. So it is like kind of dividing. One divides into two, two divides into four, four divides into eight, depending on how many mitochondria are required for their energy requirement. Okay. So in that same fashion, what will happen is like the mitochondria will divide. So if suppose there is suddenness of energy requirement, So there is like lots of energy which is required. So all of a sudden, if it is required, so new mitochondria will form. That's all. Okay, so with this thing, karyokinesis is over. We have completed half of mitosis. So now we will try to go for the next case, which is your cytokinesis. Okay, cytokinesis is separation of cytoplasm. Okay, so now the cytokinesis will happen in two distinct ways. Plant cells will have a different cytokinesis. Animal cells will have a different cytokinesis. Plant cell will have a system which we call it as a cell plate formation. Whereas in case of animal cells, they will have something called as a furrow formation. Okay. So what is the distinction between these two things you need to understand? You should understand that the plant cell has cell wall, rigid cell wall. As for, if you are seeing animal cell, no cell wall. So since cell wall is present and cell wall is absent, there are two distinct ways of this kind of cytoplasmic division. Okay, so now what we will try to see is we'll try to see how a cell plate is formed, and then we'll also try to understand how a furrow is formed. Okay, so let us try to see the plate formation technique. As far as plate formation is concerned, let us say that this is a distinct plant cell with two nucleus. Okay, the plate will start to form in the center of the cell. A small plate-like structure, a cellulosic structure will appear. This is a cellulosic plate. Is formed where? Is formed in the equatorial region. Okay. Okay. And then what will happen gradually? This will start to grow. This will start to grow in this direction from inward to outward. So as it is moving, what you will be able to see, you will be able to see that this is the cell. And then finally it will try to come and meet here. The moment it is trying to meet here, it will divide into two cells. So it will be dividing into two cells, and the cell plate will be, this is where the cell plate is there. Okay, so it will divide into the two things. So what ultimately happens in plate formation is a cell plate is formed. What is a cell plate? A cell plate is a cellulosic structure which is formed in the center. And what happens to the cell plate? The cell plate will start to grow. Okay, and what it is trying to do when it is meets the end, cell plate meets the cell wall, cell divides into two. Karyokinesis is same for both plant cells and animal cells. Okay, karyokinesis is like not very distinct for any, I mean like karyokinesis is the same in every other cell. But what you need to understand is, you need to understand that the cytokinesis part alone is different. Why it is different? Because I have mentioned it here. Why it is different? Because plant cells have cell wall. Animal cells will not have what? Cell wall. They will not have any cell wall structure. So the cytokinesis part alone will be different in plant cells and different in animal cells. So plant cells will have a structure which is called a cell plate, which is formed, and cytokinesis happens. Whereas animal cells will form a structure which is called as furrow, and then it will try to divide into two. Okay, so that is what I am showing here. So here I am trying to show how a cell plate is formed. Where is the cell plate is formed? The cell plate is formed in the middle of the cell. And what happens? The cell plate grows. In which direction? From inside to outside. The cell divides into two when the cell plate meets the cell wall. The cell plate represents middle lamella found in plant cells. And what happens to the mitochondria and plastids? Mitochondria and plastids are evenly distributed between the two cells. Okay, so this is what is for plate formation. How do plant cells form spindle fibers as they do not have centrioles? Good question. The answer is they will have some structure called as microtubule organizing structures. So plant cells will also have microtubules, which are, there are some structures which will organize those things, which we call it as MTOC. What is that MTOC? That MTOC is nothing but a cluster of microtubules which are joined together, which are organized together into a structure. Okay, so what is this MTOC stands for? This MTOC stands for microtubule organizing center. These MTOCs will be distributing the microtubules. Yeah, I think I remember telling this in the centriole class. Okay, this is what we call it as your thing. Okay, so with this thing, this part is over. So now we'll move on further. We'll move on to furrow formation. Okay, what is meant by metakinesis? This guy is asking Kanish. Okay, listen here. The chromosomes were together here. Okay, in anaphase, before anaphase, in the metaphase, what was there? The chromosome was together like this. Okay, after which they will be like separated. And what is happening is they are moving towards opposite poles. This division of movement of this chromosomes towards opposite poles from the equatorial plane is called as metakinesis. This is called as middle. Meta means middle. Kinesis means division. So the division of the chromosome is what we call it as metakinesis, which will happen during anaphase. So whenever anaphase is there, no, at that time, like whenever it is happening, this is in the middle, it will divide. That is what we call it as metakinesis. So coming to furrow formation, so what will happen? Let us say that the animal cell is not having any proper shape. It is having two nucleus. So now what will happen? This animal cell will start to show some kind of small constriction like this. So this area is called as the furrow. You're able to see some small constriction inside the cytoplasm like this. So this small part, which is like kind of like dent kind of structure that is formed, is called as a furrow. As it is growing, the furrow will start to grow. But the growth is happening inward. Where is the furrow first formed? It is formed in the periphery. From the periphery, what is happening? It is moving towards the inner side. Okay, and it is moving towards the inner side. And gradually, what will happen when the two ends of the furrow are going to meet each other? The cell will start to divide. So here there will be a nucleus, 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 again here a nucleus, here a nucleus, nucleus. Cleavage happens because of furrow formation. What we talk as cleavage, whenever a zygote is developing into an organism, that is what we call it as cleavage. The word cleavage is specifically used to indicate cell division that happens during embryo formation. Okay, so this is part of the cytoplasmic division. Cytoplasmic division is your cytokinesis. When cytokinesis is there, a furrow is formed, and then this is there. Like two cells are divided. So basically, cleavage. No, cleavage is a type of cell division. It is mitosis, which happens during what? Mitosis that happens during zygote formation, from the zygote to embryo formation, that we call it as cleavage. So a cleavage will have like lots of mitosis cycles. So every mitosis cycle will have furrow formation. So this is what is furrow formation. So you are able to understand. So furrow is formed where? In the outside of the cell. A furrow is formed. What is a furrow? A narrow constriction. Maybe you can say that. A narrow constriction. Constriction means it is a small dent is formed in the plasma membrane. A plasma membrane. Okay, not a plasma membrane. The furrow starts to grow in the inward direction. When the opposite furrows if you want, you write in the plasma membrane either side. When the opposite furrows meet each other, the cell divides into. Okay. Okay, so this is what will happen during your cytokinesis. Okay, so please, I think you are understanding. So with this thing, mitosis is over. So now what we will try to study is, we'll try to study two more things. If you have done, okay. So now what we will do is, like, you have to understand mitosis is something that we have learnt. Okay. And then you have to say some variations of mitosis. You should know something called as mitosis is the most prevalent way in which cells divide. Okay, most of the cells in your body, animals, everything, it will follow mitosis. But there are some kind of examples which are like slight modifications of mitosis. One such modification is what we call it as amitosis. And the second one is what we call it as endomitosis. Okay, so you have two modifications or variations of mitosis. The word amitosis is indicating what? Not mitosis. Okay, what is this endomitosis? It means inward in mitosis. The word endo literally means in, and mitosis is mitosis. So here, if you're like looking at the root, you should be able to understand what they are. Okay. So now what I'm going to tell is, I'll first try to indicate what is the thing. Amitosis is not exactly mitosis. In what way amitosis is different? We'll try to study. Okay. So now, what is this amitosis? In amitosis, the first thing, it is a crude cell division without what? Chromosome formation. Okay. Spindle fibers are not formed. First you write it, I'll explain it. It happens in prokaryotes. Fourth thing, Thambi, we haven't gone to meiosis yet. Prasanna, those are all things which will come in meiosis, not mitosis. It happens in prokaryotes. Okay. It may lead to variation at times. Okay. It's a simple mitosis. The distribution is so basically what you need to understand is as far as mitosis is concerned, you have clear cut distinct phases. Okay. Like you have prophase, metaphase, anaphase, telophase. Those phases are not seen here in amitosis. So when you're talking about a bacteria is a prokaryotic structure. A prokaryotic structure which is having a very simple chromosome. I mean, it doesn't even have a chromosome. It will have a circular DNA. That's all. So when the circular DNA is multiplied, no? So how it will multiply? It will try to be like kind of like multiplying, and it will remain attached like this randomly. So the genetic content is like kind of multiplying in this way. And it is like held together. Okay. So let us say that this is a point where it is held together. So here, when the cell is dividing, what will happen? The spindle fibers are not formed. So what will happen is this will randomly chumma, it will like come and divide like this. It will divide like this into two parts. What has not happened? Spindle fibers are not formed. Chromosome is not formed. Okay. The distribution is random. Randomly it is distributed. So when it is randomly distributed, what can happen? Sometimes this part of the chromosome can get into this thing. As in the distribution is not perfect. So in amitosis, what will happen is it can lead to some amount of variation at some times. Most often it doesn't, but there is a chance for variation because the distribution is completely random. Okay, so you can say this amitosis is also called as binary fission. Okay, so here there is no chance of like the genetic material where it will not get evenly distributed. So when it is all random, what might happen is some amount of randomness is there involved. So it need not be like equal in both the cells. But whereas in case of mitosis, it is always equal. And where do you see amitosis? Amitosis is seen as a mode of reproduction in prokaryotes, which we commonly call it as binary fission. Okay, are you understanding? So which means it may lead to variation. Why it is leading to variation? When there is unevenness, this will be looking like one thing and this will be looking like another thing. Some amount of what genetic nature might vary. I'm just using the word may. I'm not saying for sure it is going to happen. Okay, so it may lead to variation, whereas in case of your mitosis, it might not happen. Are we clear? So this is what we call it as amitosis. And Devadarshan, you are asking me how do viruses multiply? I have detailedly told you how it multiplies in the previous chapter, biological classification. Lytic cycle, lysogenic cycle. Are we understanding? Okay, so now the next thing is endomitosis. Karyokinesis happens, but cytokinesis doesn't happen. So what is endomitosis? It is a type of cell division where only karyokinesis keeps on happening. Whereas like in case of your cytokinesis will not at all happen. So in such a case, what will happen? It results in a single cell with multiple nucleus. Yeah, cell organelles are not there in those cells at all. It just contains inclusion bodies. Prokaryotes do not have cell organelles. Amitosis is not seen in advanced organisms. It is seen only in prokaryotes. So it results in a single cell with multiple nucleus. Okay, it should be nuclei. Okay. The structure formed is called as syntium. So what will happen? So let us say that the cell is there. This is having one nucleus. So it will undergo karyokinesis. It will lead to two nucleus. But what will not happen? Cytokinesis will not happen. Again, one more round of karyokinesis can happen. It can lead to four nucleus within the cell. And after some time, you will be able to see like multiple 3, 4, 5, 6, 7, 8. Like this, it can form. This structure is called as a syntium. Yeah, this will lead to polyploidy, right? Okay, but I don't think it is like kind of polyploidy because this is just going to have the same chromosomes which are there, but multiple copies of chromosomes are there. You cannot call it as polyploidy. You can call it as polyploidy only when there are multiple sets of chromosomes are there. Okay, here it is the same set which is getting repeated multiple times. Endomitosis is seen in plant cells mostly. Plant cells of endosperm show endomitosis. Okay, so you have this coconut water that you're drinking, no? Tender coconut water. So that is endosperm basically. We are trying to consume the endosperm. So that plant cells of that endosperm show endomitosis. This is not multiple fission. Multiple fission means many cells have to be formed. Synctium will not result in multiple fission. It will not result in multiple fission. It will just be a cell which will have multiple nucleus. That's all. Slime molds are not formed because of endomitosis. They are cellular aggregates without the wall structure. There it is all like multiple cells come in together and they are forming a collective structure that is called a cenocytic structure. Okay, so if you're talking about slime molds will be joining like this together. And then finally they will be uniting into a single structure with multiple nucleus. So here what is happening is in slime mold, you are seeing multiple cells are joined together and the walls get dissociated and then it is forming a cenocytic structure. Okay, so it is like that. Whereas in case of your endomitosis, a single cell is there dividing continuously, its nucleus is dividing continuously and it is trying to form a syncytium. So syncytium and that plasmodium are not the same. Plasmodium, which is formed in slime mold, is a unique structure which is a cenocytic structure where multiple cells aggregate together to form a single cell. Here, a single cell is having multiple nucleus. So both are distinct. Okay. Multiple fission means many cells should form. So here many cells are not forming. So we cannot call this as multiple fission. Are we understanding? Okay. So with this thing, mitosis is over. Okay, so with this thing, we'll wrap off the session. Tomorrow, we'll start with meiosis. Okay, I thought I should be able to complete it tomorrow, but I'll extend it to Monday also. Okay, I need 2 classes for meiosis. So, first slide. Second slide. Third slide. Fourth slide. Fifth slide. Sixth slide. Seventh slide. Eighth slide. Ninth slide. 10th slide. And 11th slide. Children, the doubt clarification sections are not there for biology because we are like concentrating only for JE subjects as far as doubt clarification is concerned. If you have any doubts, you can just ask in the class. Biology doubt clarifications are not wantedly kept because here this is a batch focusing on JE 1 and 2. First slide, yeah. Second slide. Eighth slide. Thank you, children. Bye. Okay, we'll meet later tomorrow. Sir, class முடிச்சிட்டேன்.